Okay, let's do something. So obviously the exam is next week. Hopefully people realize that. It will cover all the material in the cell cycle up to today, including today. No recitation today. My favorite thing is to go to recitation and see the people who didn't come to class and didn't know that there was no recitation. Just this fun little thing I like to do. Those people also don't know that they get to take the quiz next week for extra credit. There's so many pros to come to class. I mean, unless they listen to the recording memo here, but usually people are a little behind, so they miss out on those things. Am I mean? A little bit? I don't know, I like to reward the people who come to class. Okay. The quiz for this week will be available to take for extra credit at the end of next week's exam. So if you want the extra credit, read papers, think about it. If you just don't have the time, it doesn't hurt you. And I was supposed to introduce the Gene Expression Project this week in recitation. I'm going to introduce it on Monday in lecture instead. So we can at least kind of get on that. Okay. So here's the list so far of what you need to know for the exam next week. Cell cycle, all these mechanisms of regulation. I've had a few people visit me who are like, I don't know how to put this all together. It seems like a lot of stuff. So I encourage you to think of it as the cell cycle is the backdrop, and all the regulation is kind of like how it's all turned on and off. And then S phase and mitosis are things that happen within the cell cycle. And all those mechanisms of cyclins and CKIs and all that, those are all about turning the different phases on and off. And if you organize it that way, it might be easier. If you're still confused, come see me, I guess. So we talked about checkpoints. We talked about S phase. We mostly focused on DNA polymerase itself, initiation, origins, regulation of origin firing. There was a little bit of compare and contrast going on between bacteria and eukaryotic cells, priming synthesis, termination, decatenation, to the untangling, which requires topoisomerase. We talked about the end replication problem in telomerase, which would be, I guess, the answer to the problem. Then we started on on Wednesday with M phase. We talked about cohesins, condensins, regulation of M phase starting, mitotic spindle assembly, which required a little bit of background in microtubule dynamics and the concept of dynamic instability, which is useful to understand if you want to understand what's going on in mitosis at a slightly deeper level. Okay, this is where we ended, and someone, yes?
We have to know about flow cytometry and the other ways they study.
Whenever I give you methods, it's useful to know how you would use them. And so usually I give you an example of how you use them, and I might use something similar on the exam. I'm not going to say what is flow cytometry, but I might give you data and ask you, what phase of the cell cycle are these cells in, stuff like that.
So we don't need to know the steps.
Do I want you to write a protocol of an experimental procedure? Absolutely not. No, I will never make you memorize a protocol, but I will make you understand. If I could make you all understand, I'd be an amazing teacher. You need to understand the concepts. So the concepts of flow cytometry is about counting cells based on certain criteria. So if you're staining the DNA, you need to understand that you're going to have twice as much DNA in certain parts of the cell cycle, for example. Good question. I'm sure everyone appreciated it. Thank you. Okay, so this is where we ended. I got a question at the end of class by someone asking, they're like, wait a minute, so you're telling me basically catastrophe of microtubules, so that's the rapid disassembly of microtubules is what's involved in chromosome segregation. I don't understand that. How is it able to attach to the chromosome if it's falling apart? And it's basically due to this NDC80 complex. It's this protein complex that acts to attach the microtubule of the mitotic spindle to the chromosome. Here's your chromosome. This is your kinetochore here, and then there's this protein complex that anchors your mitotic spindle to your kinetochore. Okay? And basically, it acts like there's almost like a collar that's around the microtubule, and it slides back along the microtubule as this assembly of the microtubule occurs. And that's what separates your chromosomes. This complex actually gets phosphorylated by Aurora B kinase if you have incorrect attachment of your mitotic spindle to your chromosomes. The phosphorylation actually removes the microtubule from the kinetochore. And this starts to happen actually at chromatopause. Okay, so let's talk a little bit more about kinetochore proteins. So if you were trying to identify kinetochore proteins, how would you do it? I have some things up here. They may or not make sense to people, but just off the top of your head, what would be a good way to identify kinetochore proteins if you know SEMP-A is important in kinetochore assembly? What would you do? Jackson Robertson. Yeah. You can talk to a friend. So you're trying to identify, so kinetochore is made up, let me expand my question. Kinetochore is made up of a complex of proteins that all work together to bind to these other NDC80 complexes. There's a whole bunch of proteins involved. It's not just a thing, right? It's a whole complex of proteins that have a function in separating. And so the first people who were studying it said, well, we know SEMP-A is involved in chromosome segregation. That's what we know. We want to know what other proteins are involved. So how would you go about identifying these other proteins? No? Okay. Does anyone want to take a guess? Okay. Despite me having it up there. Next. Okay. So what you would do, you know SEMP-A, so you tag it with something and express it in mammalian cells. So you have it on a plasmid with some kind of tag. The reason you would tag it is so you could isolate it. You'll add on, a big one to do is maltose binding protein. You stick it onto your SEMP-A, and then you incubate it with some beads that bind maltose binding maltose, right? You incubate it with some beads and you isolate it. And what else do you isolate? You isolate everything bound to it. Okay? So you tag, express SEMP-A in mammalian cells. Why mammalian cells and not bacteria? It's different. We're studying mammalian cells? Okay. And then you immunoprecipitate or isolate it by some kind of matrix that's going to bind to your tag. So that's what you have here. Here's your cell with your tagged protein in it, and then you lyse your cells and you isolate the complex using beads that bind to your protein. So you're going to get SEMP-A and every other thing bound to it. Okay? And so that's how these were identified. And so what they did then is they identified all the proteins by mass spectrometry. Does anyone know what that is? Chemistry people should definitely know. Yep, so that's basically where you, okay, yeah.
Okay, so you put the proteins or whatever your substance is through, and it separates it based on its mass, and as it elutes, it shows you what elutes, how much of it, and then you can look at what the mass is and find it in the database.
Right, so yeah, so what it does is you put your protein, you can use it for more than just protein. You can use it to identify anything almost. And what it does is it it measures the mass, pretty much, of the protein, and so, or whatever you're doing. And so with proteins, you chop it up into peptides and you throw it in there, and the mass of each peptide is going to be specific to its amino acid sequence, right? Right? So you know each amino acid weighs a certain amount, right? And so if you have a peptide of 10 amino acids, it's going to be different than another peptide of 10 different amino acids. And so you can identify the amino acid sequence, and then you can blast it, search it against a database, and figure out what proteins you have. And it's very cool machinery. So that's what they did. So here, this is just showing a gel where they took whatever they isolated it that was bound to SEMP-A, ran it on a gel, did a silver stain, and you can see all the specific bands. And usually you have a negative control where you didn't immunoprecipitate something, so you can get, because sometimes things just tend to stick to whatever matrix you're using. And so they have all these bands, and then they cut it out and they sent it for mass spec. What you could do now is you can just send your whole mess for mass spec. You don't even need to cut out individual bands. It's called mud pit, which is kind of cool. You just identify everything in a tube. So they found all these different SEMP-related proteins. And one that they found was SEMP-N. Very creative naming. And so they decided to study SEMP-N. So remember, we're trying to study what proteins are involved in segregation of chromosomes, what proteins are involved in the kinetochore, right? So SEMP-A was used to isolate, and then they found many proteins, including SEMP-N. And so the first thing they did is look at where this protein is, right? Because if it's part of your kinetochore, should it be in the nucleus or the cytoplasm? Nucleus? Okay, there's a lot of, I'm hoping people are just sleeping, because if anyone is like, I don't know how to answer that, I need you to see me and we need to have a long talk. Because your kinetochore is where? In your nucleus or your cytoplasm? Nucleus. Why? Because your kinetochore is associated with your DNA, right? Anyways, yes.
I think mitosis.
During mitosis, your nuclear membrane does dissolve, but I'm just trying to like start somewhere. Yeah. So absolutely, during mitosis, your nuclear membrane does dissolve, but let's say you're standing at other times. It should be in the nucleus. So, should it be associated with your chromosomes? Yes, right? Should it be near SEMP-A? Probably, because you isolated it with SEMP-A. So what did they do here? They looked at metaphase and anaphase, and they used their tagged SEMP-N, which is GFP, so it's going to be green, and they used ACA, which is anti-centromere antibodies. Why? Because you know your kinetochore assembles at your centromere, right? And DAPI is for DNA, and what do they say? Well, at metaphase, it's in this line. And at anaphase, you start to separate. Is that what you expect to see? So it must be, that confirms that it's associated with SEMP-A. It confirms that it's centromere associated. It's probably kinetochore protein, right? But to truly prove that something's a kinetochore protein, you can't just show that it's somewhere. You need to show function, right? And what does a kinetochore do? What's the function of a kinetochore? Who wants to raise their hand? You can raise your hand or I can call on you. Yes? Attached to mitotic spindle, but what's the function? So yeah, so yes, that's one thing, but like give me an activity that happens when you attach your kinetochore to your mitotic spindle and you go through, thank you, yeah, you separate your chromosomes, right. Did you want to add to that?
Yeah, I was going to say that it's like mediated between microtubules and centromeres.
Yeah, so it basically connects your microtubules to your centromere, right. Okay, so what did they do? They used siRNA, which is a way to get out. Are you guys confused? Are you excited about?
I was thinking more about that, maybe it has something to do with the clarity of the cell, but I wasn't sure.
The kinetochore, that has something to do with the polarity of the cell, like in terms of what,
Like, when they say, when the chromosome segregates and separates in a bipolar direction, it's separating. Right.
that more has to do with the poles of the mitotic spindle. So what they did is they used siRNA, which is RNA that is complementary to whatever target you're trying to get rid of. So they use siRNA against SUMP-N to get rid of all of the SUMP-N, right? Because they're testing the function of SUMP-N, and they're looking to see whether or not you have normal chromosome segregation, right? Because if it's involved in the kinetochore, then if it's, you should see a defect in chromosome segregation. And so normally you have your microtubules attaching to your, actually more than anything, it's about attachment of your microtubules to your chromosomes. So normally you have your microtubules, which are green here, attached to your DNA, right? And I think it's still red for your centromeres. And then over here where you get rid of SUMP-N, look what happens. Are all your chromosomes lined up on metaphase, or are some of them in the wrong place? Look closely. I know it's far away, but like you'll notice here they're all lined up, and here there's a lot lined up, but what's going on back here? There's a whole bunch that are not where they should be, which means that the mitotic spindle did not bind, because that mitotic spindle binding is what helps with the lining up of your chromosomes at the metaphase plate. Okay, I feel like I've lost everyone today. Who's ready for Easter? Still Passover for me, but I love chocolate. Yes.
Can you repeat what you said about the vaccine?
Yes. So here what they did is they got rid of SUMP-N. Okay? The whole point is trying to prove SUMP-N is part of the kinetochore. So you need to show that there's some defect in kinetochore function if when you remove a part of it, if it's truly a part of it, right? If you didn't see any effect and it looked exactly like this, you would say, well, maybe SUMP-N isn't part of the kinetochore. But instead what you see is a number of your chromosomes are lined up at metaphase like they should be, but all over here you also have these chromosomes with these unattached kinetochores or centromeres, depending on what that, I think it's still the centromere antibody here. So they shouldn't be there. They should all be lined up. So you would say, SUMP-N must have something to do with the attachment of the mitotic spindle to each chromosome. Yeah?
Would you assume that the majority of the chromosomes would not be on the end plate because you're lacking SUMP?
Depends on the function. So the question is, would you think that most of your chromosomes expect to see them all over the place, right? It depends on the function of SUMP-N. Remember, it's not one protein involved. There's SUMP-T, SUMP-P, SUMP-N. There's some other random proteins that they isolated. So if the kinetochore is this whole machine, think of it as, that is involved in attachment of the mitotic spindle to the centromere, then there should be some kind of defect, and depending on the exact function of the SUMP-N, it might just make it less efficient. The attachment might be less efficient, so you see a moderate defect. Yes?
So, in other words, SUMP-N is associated with microtubular kinetochore attachment that is not responsible for
SUMP-N is associated with microtubule kinetochore attachment, but is not responsible. Yeah, it's not completely responsible, because if it was responsible 100%, then you should see those chromosomes everywhere. Yes?
So Dappy binds to the DNA, which is like the chromosome themselves, and then the ACA binds to the centromere in the same chromosome. Is it like the Dappy will bind to it when the chromosomes are not segregated?
Dappy binds to the DNA period. It's non-specific DNA stain. Yes?
Do we know how the cell knows how to polarize?
We'll get there as we talk about mitotic spindle. Yeah, a little bit more. Are you guys good? Are you good? Okay. I should tell you to leave because you just walked in. I'm gonna be nice because I'm lazy right now, but I have every right to ask you to leave. Okay. Okay. So let's talk about correct attachment, right? So we keep talking about how important it is to bind to your chromosomes and for your sister chromatids to stay together and for proper segregation of your chromosomes, right? So for all things to move smoothly so that you don't end up with the wrong number of chromosomes per cell, you need to have correct attachment of the mitotic spindle to the kinetochore. And there needs to be some way to regulate that and to check up on it and make sure it all works right? Make sense? So, and remember, if you don't have correct attachment of your mitotic spindle to your chromosomes, what happens? Actually, you know what I'm going to do? I'm going to ask someone. I'm going to ask Kelsey Winters. Oh, that's too bad. Kelsey, you should come to class more. Okay. It's great because I'm recording it, and ideally people listen. Okay. How about Patrick Davis? Yeah. I guess so. So if you have improper attachment of the mitotic spindle to the chromosomes, what happens? Do you just progress through cell cycle, or does something happen?
You have a rest before interphase.
So you have a rest. Not interphase, but what you mean? Anaphase, yeah. And what's that called? Does anyone remember? There are these different things that happen during the cell cycle that cause a rest when something's not right. Checkpoints. Yeah. So you actually activate the spindle checkpoint. You're right, a rest. Okay, so normally you should have this attachment where you've got equal, your mitotic spindle on this side and your mitotic spindle on this side, both attached to the centromere and pull in opposite directions. You have normal segregation. But you can have incorrect attachments. Let's say the mitotic spindle on the left, and I guess your left, attaches to both kinetochores, right? That's not right. Or let's say only one is attached, and you have an unbound kinetochore, right? So there are ways for the cell to detect this. Did I see a hand going up? Nope. Okay. So first of all, if there's an incorrect attachment, then it's destabilized by the activity of Aurora B kinase. What do kinases do? James Steckman? Oh wait, he's not here. Sam Lippincott? They phosphorylate, right. So Aurora B kinase is going to phosphorylate the kinetochore proteins, and that will cause detachment of the mitotic spindle. So the microtubule will detach, and it will allow for another try. So there's a certain amount of rounds of tries that the cell will allow to get it right, and then at some point, incorrect attachment will activate the spindle checkpoint. So if you delete, let's say you're a scientist and you're studying things and you like to knock things out to study their function, if you delete Aurora B kinase, then you're not going to get normal chromosome segregation, right? I mean, you are going to get abnormal chromosome segregation because it's Aurora B that keeps things going correctly because it phosphorylates and allows for detachment, which allows for correct attachment. If you don't get that, then you're going to get incorrect attachment, and then you're going to move through with incorrect chromosome segregation. Yes.
What hydrolyzes the Aurora B in order to, so that when it tries again, it can attach?
So what removes Aurora B induced phosphorylation so that you can attach again? some phosphatase. I have no idea what it's called, but for sure there's a phosphatase that's going to take off because once you phosphorylate, you're going. And the other thing is, yeah, because you're phosphorylating your kinetochore proteins, and that's not going to allow reattachment until that phosphate group is removed. Okay, good question. Any other questions about this? So what I'm showing in this picture here, there's the green is your microtubules, the blue is DAPI, so that's your DNA, so that's normal. And then these cells actually have now I'm questioning what it is. I always forget.
Inactivation.
Well, I think it's a deletion of Aurora B kinase because then what happens is you don't get right. So it's inactivation of Aurora B kinase, which results in incorrect assembly of mitotic spindle to the chromosomes, so they're all over the place. You can see your mitotic spindle, you've got some microtubules out here bound to some chromosomes, some are out here bound to some chromosomes. That's not right. Yes?
So not having Aurora B would not activate the spindle?
It should activate the spindle checkpoint point. But forget that this is at the point before that happens where you are trying to detach chromosomes. So technically, let's say forget spindle checkpoint for a minute, and there is a chance that deletion of Aurora B might affect the spindle checkpoint in a way, rather than activating it. It might cause a defect in that checkpoint. I'm not 100%, but it will definitely allow for incorrect attachment of the mitotic spindle, and then most likely incorrect segregation. Yep. Any other questions? Yes.
Makes sense already. What activates Aurora B?
What activates Aurora B? Do I go into this in this one? The cell has a way to detect the tension of the microtubule bound to the kinetochore. And so if incorrect tension is detected, then Aurora B will come and phosphorylate. And so you can imagine the tension from appropriate attachment is going to be very different than the tension from one spindle attaching to both sides, right? Yeah, good question. Yes?
So does the spindle checkpoint occur after Aurora B works and then they still aren't attached?
Yeah, so Aurora B will allow for reattachment, and at some point the cell says, okay, this isn't working right, let's activate the spindle checkpoint. Yeah.
So Aurora B isn't essentially what activates the spindle checkpoint, it's if it's like not working with the cell.
Right, so think of it as two steps. One is allow for a redo, and the other is, okay, let's stop everything.
Okay.
So Aurora B is the redo. Yes?
So then, I just want to hear, Aurora B like takes it off and then has another chance to put it back on.
Right. Aurora B phosphorylates, allows for detachment, and then that allows for reattachment. Yes?
Does this happen if there's only one spot that's not working?
Does it happen if there's only, so if one chromosome is incorrectly attached but the others are all okay? Yes, because even one chromosome not being properly attached, or one sister chromatid not being properly attached could mess everything up if allowed to properly segregate, or allowed to move to anaphase. Yes?
Based on this question, so will the Aurora B just go to that one misconduct?
Yes, so that's what I'm saying is it's sensing the tension of the microtubules bound to, the tension within the mitotic spindle. And so each point of the microtubule bound to a kinetochore is a point of tension, right? So it can detect them all individually. And you're only going to phosphorylate what needs to be detached. You're going to leave alone what's fine. Yes?
Would there be like increased amount of Aurora B kinase if the cell was sent and knocked out?
Would Aurora B,
Or is it not even active?
so the question is, will Aurora B kinase be upregulated in cells with no sense N? Well, I like the way you're thinking, and I guess that's very logical and plausible that there's probably some kind of feedback. I don't know how quickly gene expression happens. You know what I'm saying? Because this is all happening in an extremely short period of time. And it's also during mitosis where your chromosomes are highly condensed, so you're not likely to be having a huge amount of gene expression. Yes, but if there's a way to regulate Aurora B activity, I would say you would see a higher level of Aurora B activity in those cells. Yeah. Any other questions? Okay, I'm getting great questions today. That means you guys are not asleep. For a minute, I was really worried that like you had all turned into zombies. My kids, when they watch TV, they just like zombify like that. And so, and I start to see that look. I'm like, either I'm as good as Dora, or you guys are thinking about Dora, not me. Okay. So I was going to say, let's discuss how Aurora B prevents spindle attachment. We've kind of just discussed it, so we'll keep moving in the interest of time. Okay. So Aurora B did its thing, and it didn't work, right? So Aurora B phosphorylated. You had many rounds of trying to reattach properly, and it didn't work. So then what happens is you get spindle checkpoint activation, and this inhibits APC activation by inhibiting CDC20. So this is going to require you to stop and think and say to yourself, ooh, what was APC? What does APC do? What does CDC20 do? And what? So we'll talk about it for a second. Right, so you've got your chromosomes, and they're sitting here, ready to separate, and you've got your kinetochore, and you've got your cohesins, and you've got this other protein complex that called Shugoshin, which helps to protect your cohesins. And normally, you get activation of APC by MCDK. And that degrades all this, and then you get chromosome segregation. But if things aren't attached properly, which is what you see here, your mitotic spindle is only attaching to one side, then CDC20 is inhibited, which, remember, CDC20 activates APC, and APC is the ubiquitin ligase that ubiquitinates and degrades all these things. So you need, well, not all these things, let me take that back. It ubiquitinates and degrades MCDK and M-cyclin, securin, and so you got securin. Remember, those need to be cleaved by separase, and Shugoshin, which I haven't talked too much about yet, which helps to protect the cohesins from being prematurely degraded. Those both are degraded by APC. So if APC is turned off, that can't happen, so you can't progress to anaphase. So spindle checkpoint activation is inhibiting progression to anaphase by turning off APC. Okay? Faulty spindle checkpoint actually is seen in cancer frequently, and that's because, and you end up seeing this genome instability because you still can segregate your chromosomes even when you have improper spindle attachment, and that's why you start to see aneuploidy in cancer. Any other questions? Thoughts? Everyone's excited to take an exam next week? Okay. Yes?
So cancer cells, what exactly allows it to progress to
Defects in the spindle checkpoint. So many, like Aurora B kinase, I don't know, so many things. Well, one thing that we're going to talk about now is MAD2. And MAD2, it binds to unattached kinetochores and actually changes its conformation, associates with MAD1 and some other proteins, and binds to CDC20, which then inactivates APC. So if that doesn't work, if this whole system doesn't work, APC is then still on, and then you can still move forward even with incorrect attachment. So there's actually many different proteins that are involved in this, and a lot of them have mutations in them in cancer. So MAD20. So here's a cell, well, you're really just looking at the mitotic spindle and chromosome. So green is tubulin, blue again is DAPI, the chromosome stained. And you can see you've got your mitotic spindle attached, your chromosomes are all lined up at metaphase, and then you've got this guy over here. I feel like I must be boring people today. People keep getting up and leaving. Is it boring today? A little bit? No? Feelings are hurt. Okay. I've watched like 3 people get up and walk out. What is it? Is it registration day? Not yet. People get crazy on registration day. Or like leaving and registering in class. I understand. Okay. So this right here is MAD2. See that red dot right there? So MAD2 binds to the unattached kinetochore because this kinetochore here is not attached to the mitotic spindle. Notice there's no MAD2 up here. These are all properly attached. Okay? And then what ends up happening, let's see, did I put in a slide? Oh, right. So this is my reminder slide for you guys. Remember, CDC20 is required for activation of APC. So if you've got no APC, you've got no anaphase, right? So you've got inactive APC, and then CDC20 comes along and activates it, and it does a whole bunch of things, including degrading securins, so separase can come along and degrade the cohesin. And MAD2 actually binds CDC20, so then this can never become activated, this can never become degraded, this can never be separated. Okay, so that's one thing that happens during the spindle check, when the spindle checkpoint is activated. So normally, if all goes well, right, so the mitotic spindle attaches appropriately, separates, degrades cohesin, and the cell cycle moves on, and mitosis moves on, and we're all happy. Something to just take note of the fact is that separase is inactive until anaphase, and that's highly regulated, and it's regulated by multiple mechanisms. One is what I keep telling you guys about securin. Securin binds and it keeps it inactive. There's also phosphorylation that keeps it inactive, and then there's also these proteins called Shugoshin, which bind to cohesins and protect them in case for some reason there's something's mutated. Let's say securin's mutated and separase is always active for some reason and wants to cleave the cohesins in the absence of APC activating it. Well, you still have another protection by the Shugoshin complex. So it's obviously, you can see things that are important in the cell, you need to have multiple levels of regulation to prevent things from happening when they shouldn't. And obviously a really important one is you don't want to move to anaphase if things aren't right, because your next cells will be a mess if they don't have their appropriate chromosomes, right? Okay. APC activation, so this happens towards the end of M phase, and it allows for sister chromatid separation, right? So you've got your securin separase, which is inactive, and your metaphase chromosomes are held together, and then when you activate APC with CDC20, then you get ubiquitination of mCDK, also known M-cyclin, which is also cyclin B, securin, and the Shugoshin, which allows for separation. Okay, we're gonna make it. Only 10 more minutes. You guys are okay? Everyone's okay? Get a question? What? Okay, so what order does it happen? That's so you've got your mitotic spindle is going to attach to your centromere long before it's like prometaphase when that starts to happen. And then your mCDK, your rises in your mCDK activity will then turn on APC, and it's all regulated by APC turning on, pretty much. Because once that's on, it can cleave all these things. It can activate separase, and then that's a big thing for that transition from metaphase to anaphase. Does that answer it? Yep. Okay. Fine if you need me. Okay, any other questions? Okay. Okay, so once the mitotic spindle is appropriately attached to kinetochore, cells can proceed through anaphase. And that happens in two phases, anaphase A and anaphase B. In anaphase A, your kinetochore microtubules are shrinking, so that's that whole NDC80 complex and the disassembly of the microtubules, and they're shrinking and moving away, right? So here's your kinetochore microtubules, so the ones that are attached to kinetochores, and they're going to shrink back towards the centrosomes. So it looks like that. But you still have these guys here. And so what are those guys doing? Well, so it's this microtubule flux or treadmilling. I don't like using the treadmilling with microtubules. It feels like it's more actin, but whatever. That's what it says in the book. It's that microtubule disassembly, which is actually involved with a bunch of proteins that help to encourage this disassembly. That is what is going to move the mitotic spindle back and separate the chromosome. Okay? The kinetochore is what keeps the chromosome attached to the spindle despite microtubule depolymerization, because it's got this complex, this protein complex here attached to the kinetochore. So then there's, yep, sorry.
So the degradation of tubulin occurs at the end towards the kinetochore, not the end towards the.
Right. Yeah. Yeah. So the question was, or the clarification was, the disassembly of the microtubule occurs at the plus end, or the part that is attached to the kinetochore, not the part that's attached to the centrosome. The centrosome actually is kind of, you can think of the centrosome as kind of like a seed where microtubules grow from. So you don't ever really have disassembly at that end. Stop me if you've ever seen it before. But the centrosome actually contains many molecules of a protein called gamma tubulin ring complex and gamma tubulin. And gamma tubulin is like the seed that starts the growth of the microtubules. So it's always at the other end, attached to the chromosome. Yes?
You're gonna go into this like more in depth, or is this just like because I don't see how it detaches from that end without coming off.
Right. So that's what this whole complex is. This protein complex.
Yeah.
It's so it has like a it's almost like a collar. Think of it as a collar that moves, that slides down. So this whole thing slides down the microtubules towards the centrosome.
How does it slide?
I don't know. Using a motor protein. I don't know. I really don't know. For sure don't know. But we can look it up. But it does move, the whole complex moves this way. So you're right. So that's always the confusing thing. You're like, wait, your microtubule is disassembling. How are you still attached? And it's based on this protein complex here. Yeah. Yes?
So the movement is of the complex, not of the microtubule.
The microtubule is falling apart. It's catastrophe that's happening. And so it's just not going to be there. So all these little subunits are going away and it's shrinking.
But what I'm saying is that complex, the chromosome, is what's moving, not the microtubule, but that complex.
Right, the microtubule itself, well, it's moving because it's disassembling.
Yeah,
But it's not actually, yeah. So this is a wording thing, right? Right, so there's confusion about moving versus disassembly, but this whole complex is attached to the microtubule and sliding towards the pole of the mitotic spindle as the microtubule is disassembling. Okay? Yes?
There's like an error. It doesn't slide.
So the question is, if there's an error in this process, is there any way to fix it, or what happens to the cell? Because you've passed your spindle attachment checkpoint. My guess is there is something, but I've got no idea what it is. I mean, yeah, because this is, and there's probably many mechanisms of regulation surrounding this process because it is so critical to the survival of the species, right? But I don't know exactly what it is. We can do an extra credit on that if you want. Who wants to do an extra credit? Or who's like, no, I need to study. Well, then don't do the extra credit. Okay. So the question is, is there any sort of checkpointy like mechanism of regulation that's going to either stop things or allow for repair if some part of this process of chromosome segregation goes awry? Either the proteins that encourage catastrophe don't work, or some of the motor proteins involved aren't working. I don't know. Maybe the kinetochore. I almost feel like a lot of those proteins are involved in the just assembly of the mitotic spindle, and you should see a defect earlier, but I don't know. So let's make it an extra credit. So tell me if you can find any research into regulation of chromosome segregation once you pass the spindle assembly checkpoint. Okay? And of course, by tomorrow midday. Okay, so anaphase B, we can do it. 3 minutes to go. Spindle poles are pushed apart during anaphase B. So anaphase A is about that catastrophe of those microtubules, the kinetochore microtubules, and the separation of the sister chromatids. And part B is about pushing the spindle poles apart through two mechanisms, both of which are motor protein dependent. Remember we talked briefly about motor proteins. If you go back to the slide earlier where I showed the different motor proteins attached to the mitotic spindle, it'll start to make even more sense. So you've got your interpolar microtubules. These are these middle ones here that are kind of looking like they're attached to each other. They're really attached by kinesin 5, which has two motor domains and causes them to slide against each other, so it's pushing your mitotic spindle apart. And in fact, these microtubules are growing. They're actually not disassembling at all. They're actually growing because you need to keep pushing the mitotic spindle apart. And then you've got your astral microtubules. These are the ones that are pointing out this way. You can think astral because it kind of looks like a star, right? Remember, these ones are pushing against the cortex of the cell, so against the plasma membrane of the cell. And they actually have motor proteins that are attached to the cell cortex that walk along these microtubules, and that's also helping to pull the mitotic spindle apart. Why do you need to pull a mitotic spindle apart? Don't we just need to pull apart chromosomes? Why would you want to pull a mitotic spindle apart? Go back to this picture. I like that one. So there's A, where you just have the chromosome segregating, and here's B, where the mitotic spindle is pushing apart. Push all a whole bunch of. You need to divide the cell, right? Remember, this is part of a larger process called mitosis, right? So you're going to get telophase and then cytokinesis. So you need to push everything apart, so then you can have the next part, which is, I think I'm ending here with telophase, where you've got everything, all your chromosomes on either side, your nuclear envelope starts to reassemble, your mitotic spindle disassembles, you go back to your interphase microtubule network, and your chromosomes start to decondense. And you should be thinking, well, how does this all happen? Don't forget, all of this was controlled by phosphorylation by MCDK. Now MCDK is gone because APC actually degrades it, and then you get upregulation of phosphatases that start to dephosphorylate. And this is, I believe, where we end. So happy studying. There is a quiz this weekend or next week. Enjoy.